Hi everyone, welcome to ACE Achievers Dental Academy. I am Dr. Bhavna Pant, MDS Oral Pathology. Today we will be discussing NEET pattern 2022 questions of dental anatomy and oral histology. So to begin with our first question. So first question was new gene involved in odontoblastic differentiation. So the options were PHX1, P53, PLAG1 and MAP1B. So remember that when we are talking about PLAG1, so remember PLAG1, the mutation, this question has come before in the exam, it is responsible, the mutation in this gene is responsible for pleomorphic adenoma, right? Okay, and when we talk about the gene P53, P53 is also known as guardian of the genome, and it is mainly responsible for controlling the cell division and death. Okay, so the mutation in this gene is mainly responsible for cancers. Fine. Apart from that, when we talk about dentinogenesis in the new organs, it talks about two major genes. Okay, one gene is your MAP1B, which is responsible for odontoblastic differentiation, as the question is asking. Apart from that, there is another gene which is PHEX. Okay, so this gene is responsible for dentine mineralization. Okay, so this question, so the correct answer here is MAP1B. So this question has come, maybe next time they might ask about the PHEX gene, so you can remember this. Okay, so I hope this question is clear to you all. Next question is type of collagen present in Von Kopf fiber. So remember, Von Kopf fibers are present in mental dentine. Okay, they are present in the mental dentine. As you all know, the parts of the dentine, we have the primary dentine, we have mental dentine and the circumpulpal dentine. So remember, in the mental dentine, we have Von Kopf fibers, which are large diameter fibers, which are mainly formed from type 3 collagen. Okay, apart from type 3 collagen, there is also presence of fibronectin. Okay, so this is important. One more important thing about Von Kopf fibers is that they are ergyrophilic in nature. So what are ergyrophilic? Meaning they can take a silver stain. So they are silver stain loving and gives black color. So when you visualize it under the microscope, they appear as black in color. I hope this question is clear to you all. Okay. Moving to next question. So here the correct answer is type of collagen present in Von Kopf fiber is type 3 collagen. I hope it is clear. Next question, that is question number 3, that is organic inner lining surrounding dentinal tubules is called. So when we talk about the organic inner lining surrounding the dentinal tubules, the correct answer here is your lamina limitans. So what is lamina limitans? As you can see, if this is your dentinal tubule, surrounding this dentinal tubule, we have a very thin organic sheath. Okay, you can call it a sheath or a membrane, which is organic in nature, first thing, and the second thing, it can only be visualized under electron microscope, not under the regular light microscope. So, and also apart from that, you also need to understand the other options. So when we talk about lamina lucida and lamina densa, remember lamina lucida and lamina densa both are the parts of basement membrane. Both are the components of the basement membrane. Okay, and when we talk about lamina dura, as you all know, it is a thin layer of dense cortical bone which lines the root of the teeth. So this you see in the radiograph. So under the radiograph, it appears as a radio opaque line surrounding the tooth socket. I hope this is clear to you all. Okay, so here the correct answer will be lamina limitans. Okay, moving to next question. Which tooth has primitive morphology and does not resemble any other tooth? So this is a very, so whenever we talk about dental anatomy, the answers are very straightforward. Okay, so here important things for you to remember is that the primary maxillary first molar resemble permanent premolar. The primary maxillary second molar resemble permanent maxillary first molar. The primary mandibular second molar resembles your permanent mandibular first molar, and your primary mandibular first molar does not resemble any other tooth. Okay, these are facts which you have to remember. Okay, so here if you see the option, so when we talk about the primitive morphology, the tooth having primitive morphology and not resembling any other tooth, the option being maxillary third molar. So maxillary third molar may or may not resemble the maxillary molars. Maxillary primary first molar, maxillary permanent first molar, and the third option is mandibular primary first molar. And as I've already told you that the mandibular primary first molar has a primitive morphology and does not resemble any other tooth. So here the correct answer will be mandibular primary first molar. Okay, so next we will move to our next question, that is plexus of Rashko is present in cell-free zone, cell-rich zone, pulp core, or odontoblastic layer. So remember the pulp has 4 histologic zone. You must have read first is the, as we go down from the dentine, so first is the odontoblastic zone, second is your cell-free zone of wheel, third is your cell-rich zone, and fourth is your pulp core. So remember that between the cell-rich zone, as you can see in this picture of the cell-rich zone, okay, and below the cell-rich zone, actually here we have the pulp core. So between the cell-rich zone and the pulp core, we have something known as nerve plexus of Rashko. Okay, or plexus of Rashko, which is mainly a plexus of myelinated nerve. So remember here the plexus of the nerves which are present are myelinated. So as these nerves move from this plexus of Rashko towards the cell-rich zone, and from the cell-rich zone to the cell-free zone and to the odontoblast, these nerve plexus becomes unmyelinated. So till the time they are in this plexus region, they are myelinated. I hope this is clear to you all. So plexus of Rashko. Here when we talk about the plexus of Rashko, so remember the plexus of Rashko is present in the cell-rich zone because they are present between cell-rich and pulp core, but they are more towards the cell-rich zone. So we call it to be under cell-rich zone. Okay. Next question, the tooth morphology varies according to tooth. Which cusp of the maxillary second molar is the smallest? So the 4 options are mesiobuccal cusp, distobuccal cusp, mesiolingual cusp, or distolingual cusp. Okay, so as you can see here, remember the maxillary, when we talk about the maxillary second molar, it has 2 types of cuspal morphology, the cuspal anatomy. That is, one is the rhomboid, which you can see here. So this is the rhomboid morphology here. Okay, so this is the rhomboid morphology, and the other. So this rhomboid is the 4-cusp type. Okay, the other is the 3-cusp type, which will have a heart-shaped occlusal anatomy. So now 2 questions keep on coming. So first question, which has already been asked, which is the smallest, which is the smallest cusp of the maxillary second molar. So remember, out of the mesiobuccal cusp, mesiolingual cusp, distobuccal, and the distolingual cusp, this distolingual cusp is the smallest cusp. The second question which keeps on coming is that which of these cusps does not form part of the maxillary molar primary cusp triangle. So this is your maxillary molar primary cusp triangle. Okay, so this maxillary molar primary cusp triangle is involving your mesiobuccal cusp, distobuccal cusp, and mesiolingual cusp. Okay, so this distolingual cusp is not a part of the primary cusp triangle. So this question has also been asked. Okay, so both the questions are clear to you all. So if a question comes, the tooth morphology, which varies according to tooth, which cusp of the maxillary second molar is the smallest? The clear answer is your distolingual cusp. Okay. Next question. Myoepithelial cell has contractile properties. Apart from that, it also has tumor suppressor function. Which protein produced by the cell has these functions? So this is a very, I would consider it to be a little difficult question for you all. But since the question has come, so you have to understand that when we are talking about the tumors, okay, the cell proliferation. So remember the matrix metalloproteinases. Okay, just understand the matrix metalloproteinases are responsible for tumor growth as they are responsible for degrading the matrix barrier. Right. So when we talk about the fibroblasts, so as you all know, fibroblasts are the one which are producing collagen as well as degrading collagen. Right. So the fibroblasts, how they degrade the collagen, that is by the production of these matrix metalloproteinases. They will eat up the collagen. Okay, but in the tumor progression, this function is basically negatively impacting the tumor proliferation. Okay, it is increasing the tumor proliferation because it is degrading the matrix and allowing the tumors to move. For example, we take the example of squamous cell carcinoma. Right, the epithelial cell, when they break and enter the connective tissue, and from there they proliferate and move deeper into the connective tissue and into the surrounding structure. If this collagen barrier is present, it will restrict these tumor cells to proliferate and move into the surrounding tissues. But matrix metalloproteinases, they actually eat up this the whole the matrix structure and allow the tumors to proliferate. So and apart from that, they have angiogenesis potential also, right? So these two are the main functions of matrix metalloproteinases. So what the myoepithelial cells are doing? So myoepithelial cells are producing something known as TIM. TIM stands for tissue inhibitor of metalloproteinases, right? So there are mainly 4 types of TIMs, that is TIM1, TIM2, TIM3 and TIM4. So TIM1 is the one which is majorly produced by myoepithelial cell, and these TIMs, they are mainly responsible for blocking the activity of the matrix metalloproteinases and hence does not allow the matrix to degrade. And that is how they are helping in tumor suppressor function. Okay, so I hope this is clear to you all. The other two options, the PRP and the histatins, they mainly have antimicrobial action of the cell in the saliva. Okay. So coming to next, so the correct answer here will be TIM, that is tissue inhibitor of metalloproteinases. Okay. So question number 8. A transient structure which plays an important role in shape of crown during formation of tooth. So remember here, when we are talking about a transient structure, so transient structure is something which is coming for a very brief period of time. Okay, so here we are talking about the stages of tooth development. Okay, the bud stage, the cap stage, the bell stage. Okay, so all these are the stages of tooth development. In between, there is a transient structure which comes and goes. So we will start with the correct answer first. So remember this transient structure, you have to remember that it is enamel knot. So enamel knot is a cluster of non-dividing epithelial cells which is visible in a section of molar cap stage tooth germ. Okay, so it is said to represent an organization center that orchestrates the cuspal morphogenesis. Okay, so that determines the cuspal morphogenesis, meaning the shape of the cusp. Okay, so they are expressing, so how do they do it? So they are expressing a lot of signaling molecules. Okay, we will not get into the details of signaling molecules here, but so for just simple, in the simple terms to to understand, just remember they are expressing a lot of signaling molecules and determining the cuspal morphogenesis. So when we say the cuspal morphogenesis, meaning morphology meaning the shape. Okay, so the shape is determined by the enamel knot. Okay, so this enamel knot in certain histological sections. Okay, so in some histological section, this enamel, so here in this structure, you can actually see this cluster of cells. So the cluster, this is the cluster of cell over the future cusp tip. Okay, so this cluster of cells, when proliferate deeper into the stellate reticulum. Okay, so remember this is known as enamel cord. And when this enamel cord moves further and touches the outer enamel epithelium, so this is your enamel, inside is your inner enamel epithelium, and this outside is your outer enamel epithelial layer cells. Okay, so as this moves from the inner enamel epithelium towards the and touches the outer enamel epithelium, this is known as enamel septum. And when it touches the the outer enamel epithelium, it forms a depression. Okay, the outer enamel epithelium will actually show a depression which is known as enamel navel. So remember, all these are the transitory structures, and apart from that, enamel, so here you can see, now you know about enamel cord, enamel navel, and there's something known as enamel niche. Enamel niche is more of an artifact which comes in between, as it is also a transient structure which is seen in which can be seen in different histological sections. Okay. So the correct answer here is enamel knot. Next important question: mesiolingual developmental groove is present in. So remember, as I already told you, the dental anatomy questions are very direct. Okay, so you need to know the facts. You need to know the occlusal anatomy. There is nothing much to understand but to remember. Okay, so when we say the mesiolingual developmental groove is present in, so the correct answer here will be mandibular first premolar. Okay, so as you can see in this picture, so this is your mesial side, this is your distal side of the mandibular first premolar. Okay, so as you can see, there is this is your lingual, right? Since it is a 2D structure, you can visualize the 3D out of it. So as you can see, on the mesiolingual side, there is this developmental groove. Okay, this developmental groove is characteristic of mandibular first premolar. This is a direct question to remember. Okay. Similar very important question in which the students keeps on getting confused is your mesial marginal developmental groove is present in. So the previous question was the mesiolingual developmental groove, and the next question here is mesial marginal developmental grooves. Okay, there are two main groove questions which keeps on coming. So for one thing you have to remember, what is a groove? Groove is a depression, right? So when we say mesial marginal developmental groove is present in, okay, first the correct answer here is maxillary first premolar. So as you can see the occlusal anatomy of the maxillary first premolar, you can see this is the distal marginal ridge here, and this is the mesial marginal ridge. So as you all know, ridges are the elevations, and over that mesial marginal ridge, you can see a line passing this mesial marginal ridge, meaning the line is cutting the ridge, and that line which is cutting the ridge is basically a groove, and that groove is known as mesial marginal developmental groove because it is cutting and crossing the mesial marginal ridge. I hope it is clear to you all. So here again, this is a direct question. You have to remember that mesial marginal developmental groove is present in which tooth, right? So the correct answer here will be maxillary first premolar. Okay, so I hope we are clear with this question as well. Next question: keratinocyte cell migration from basal layer to spinal layer process is known as. So keratinocytes, as you all know, they are the major cells which are seen in your epidermis and the epithelium. We have the keratinocytes and the non-keratinocytes. Keratinocytes are the major cells. What are the different layers? So what are the different layers when we see in the epithelium? So we have the stratum basale, the lowermost layer present towards the basement membrane is your spine, the basal layer, that is stratum basale. Then you have the spinous layer. Over the spinous layer, you have the granulosa layer. Over the granulosa, you have the corneum layer, right? This is how it goes. So remember, as the cell is migrating, so when we talk about the basal cells, okay, so as you all know, the basal cells have stem cell potential. Okay, so since they have the stem cell potential, when mitosis takes place, okay, it is completely up to the cell whether the cell wants to stay in the basal cell layer or wants to move up into the next layer, that is your stratum spinosum. Okay, so when the cell decides to move from the basal into the next layer, that is your spinous layer. Here actually it is written basal layer to spinal, actually it is spinous layer. So what happens? It is committed. Okay, it becomes committed to the biochemical and the morphological changes. Okay, so when it becomes committed to these changes, this process is known as differentiation. Okay, so just remember, so here when we are talking about the cell moving from the basal to the immediate next layer, right, it will lose its stem cell potential and it will be doing the next function. The whole structure will change. So this process is known as differentiation. But if we say, if the question would have asked moving from the basal layer to the corneum, to the topmost layer, okay, the keratinocyte cell migration from basal layer to the corneum, this is known as maturation. Okay, and the movement of the cell, movement to be specific, from one layer to another is known as migration. Clear? So just remember all these important terminologies and what the question is asking. Okay, so here this says about basal to spinous layer, to the immediate next layer. So that will be your differentiation. I hope this is clear to you all. Okay, so answer here will be D, that is your differentiation. Next question is tensile strength of the bone depends upon. So options are hydroxyapatite, calcium, collagen, and phosphorus. So remember, the tensile strength of the bone depends upon both hydroxyapatite crystals as well as collagen fiber. But better option here is collagen fiber. Still, we are doing research on this question, and we will be putting up the correct answer, final correct answer in the book. So with this, we are done with our dental anatomy and histology part. So thanks for watching.